I'm going to be presenting. I'm the president of Lighthouse Lab Services. I founded Lighthouse 19 years ago, and I have Dr. Corti with me as well, who's one of our PhD laboratory directors, who's going to be speaking to the more technical and scientific natures of some of this and the validation and what it would look like to validate monkeypox in your laboratory. And we'll go ahead and we'll get into it. So what is monkeypox? I'm sure everybody's hearing about it. I thought I'd just grab a couple key facts to make sure we all had appropriate understanding of that. It's not brand new. It was discovered in 1958. It was discovered in only animals. First human case showing up in 1970. It's rare. Same family of viruses as the variola, which is where smallpox resides. It's the same virus that causes smallpox, but the symptoms are much milder, and monkeypox is rarely fatal. Not related to chickenpox, which is a common misconception. Prior to 2022, almost every case of monkeypox occurred in Central Africa, or if it did show up anywhere else, it was linked to people that had traveled there or come into contact with someone that had traveled there or was from there. So it was pretty isolated to that region for a very long time before we started to see it spread. It is spread through close, intimate contact. I don't see it spreading or doing anything like COVID has because of that. It's not something that can be very easily spread. It is, only through that close, intimate contact that occurs. There is some research because there's been claims made that maybe it can spread through respiratory or non-symptomatic patients. We're not sure if they are able to spread it. So that is being researched and is still a TBD. The U.S. monkeypox outbreak, as of the 26th, the U.S. leads the world in total confirmed cases with a little over 24,000. I don't believe that's representative of the fact that we have the largest percentage of monkeypox cases in the world. I think we just have some of the most testing of that. And so Central Africa is not known for having a robust laboratory testing network. We've seen that throughout COVID. And I think that's true here as well. So I think that's where majority of it still resides, but we are seeing it spread worldwide with 106 countries now showing positive cases for that. Still, these numbers, if you're used to tracking COVID numbers, are going to seem pretty small, and they are. 197 new cases is the 7-day average in the U.S. All of this data was pulled from the CDC. So still at a small level in comparison to COVID, and I think that's something to keep in mind as we look at this. So, kind of what is the opportunity for your lab? I would say if your expectations are anything like this could be the next COVID play, I think that that's inaccurate, and I would kind of reduce those expectations. I do think there is still opportunity though. So if your lab was performing COVID PCR testing, especially on an open system, I think you're going to have most of the pieces that are needed in order to validate that. And so you have staff that knows how to perform PCR testing now. You have instrumentation that maybe has capacity to be able to do that testing, as we've seen COVID start to fall off. I think one of the best use cases I've seen for bringing this in is to open up new markets and new customers, right? So there's only a few laboratories in the country that have validated this. I want to say it's like less than 12 that have it validated in-house for testing. And so gives you a really good opportunity to stand out. And if you have been trying to win business from local health system, it could be a way for you to get your foot in the door because they're probably sending that testing to maybe a LabCorp Quest or one of the ARUP, some big reference lab. And if you were a regional player that was able to offer it, I think you'll probably be the only regional player that has that on the test menu. And maybe you're able to use that to open up that relationship to start serving other areas. So that's where I would see the opportunity being. It also gives your sales team kind of something unique to pitch, which I know sales reps are always looking for that edge, or how can they stand out, because they're usually not able to stand out based on price. So everybody talks about turnaround time and quality, and they've probably beat that drum pretty hard. And so if they're able to go in and say, hey, we wanted to let you know, doc or health system, that we now are able to do monkeypox testing, if that comes up, it's at least something unique to add to that conversation and maybe move that conversation forward. Some of the cons about it, and this is still a lot of opinion in here, I guess, but the volume is low, and so I don't think it's going to be a really strong long-term play. I don't see this even having the legs that probably COVID has. And the validation samples at this time can be fairly expensive. We've seen them range from $100 per positive monkeypox sample up to $5,000 per positive. We do have access to some of those if it was something you're looking to validate. So we've been able to do that for our customers where we validated monkeypox testing, but they were not cheap. I think they're going to get cheaper very quickly as we see more and more labs validate this and have a biobank of samples that they're holding. And we're doing that as well and increasing our inventory of those. So I'm going to turn this over to Dr. Corti so he can start getting into some of the CDC guidance, FDA guidance, and some of the validation requirements for adding monkeypox testing into your lab. So Dr. Corti, I'll let you take it from here.
All right, thank you, John. So as John alluded to briefly, I'm just going to discuss the current state of the FDA's monkeypox guidance, including options for validation and how to prepare your lab to receive and run monkeypox samples. So there are 3 classifications currently for monkeypox assays: the cleared assays, emergency use authorization, and laboratory developed tests. So the only cleared assay is the CDC's test, and this is only for use in CDC designated laboratories, so large public health laboratories and a few major research facilities like Mayo. For EUA options, Quest is currently the only manufacturer to receive an EUA to date. They did earlier this month, and their assay will only be available to Quest designated laboratories. The FDA is currently accepting EUA applications from organizations with known manufacturing capabilities and experience, and you have until October 13th to file an intent to submit an EUA with the FDA for priority review. So the FDA is going to want to see a description of the technology, manufacturing capability, ramp-up times, test throughput, and then timelines for validation, and then the full EUA submission, including any validation data that you have at this time. Now, the FDA has taken lessons learned during COVID and is gearing the EUA process for monkeypox toward manufacturers and not toward individual laboratories. Most individual laboratories will be better served with the LDT process rather than the EUA process. So any qualified high complexity laboratory can validate and offer their own monkeypox assay at this time under the current guidance. So the specific wording, obviously there's some caveats, but the specific wording says, at this time, the FDA does not intend to object to the offering of monkeypox tests developed and performed in a CLIA certified laboratory that meets the requirements to perform tests of high complexity as long as they meet these 5 conditions. The test has to be run at a single location. It has to use PCR technology, so no antigen tests or serology at this time. It uses lesion swabs collected by a healthcare provider. So lesion swabs are currently the only method for diagnosis of monkeypox infection, and any other specimen type will require additional review. And the test has to be appropriately validated, and we're going to come back to that one. And finally, the laboratory has to notify the FDA of the validation within 5 days of offering the test. Now, number 4 is probably the one we should go back to and discuss in a little bit more detail. The FDA is giving a great deal of freedom to the laboratory director in deciding how the validation should be conducted. This is actually a bit of a point of confusion because the FDA has released a great deal of guidance for validating monkeypox assays, but the majority of what's being presented is geared toward EUA submissions and very little directly relates to LDT validations. Now, this is to remove barriers to allow increased testing capacity in laboratories that have the ability to conduct the testing and also have the patient population to merit offering testing. In addition, the guidelines around the EUA modifications favor increasing test capacity as well. So as more manufacturers receive EUAs, modifications to the EUAs will be treated less stringently than they were during COVID. So if you want to run an EUA assay with a different extraction method or matrix or a different platform that was included in the EUA, as long as you do a full LDT to verify performance and then you alert FDA within 5 days of going live on the modified assay, you can run the assay under the EUA with no FDA review. So the method we're discussing here for monkeypox is well within the expertise of any lab currently running COVID testing. So if you have a need, you can absolutely offer monkeypox testing to your providers and your patients. Now, regarding the validation itself, the FDA has released 2 templates for conducting and submitting EUA applications, and we can glean a lot of information from them, but they do not necessarily apply to LDT validations at independent labs. Again, lab directors have a great deal of freedom in how they conduct the validation. We've assisted several labs in offering monkeypox testing, so please let us know if you have any questions or if we can be of assistance in any part of your validation, whether it's getting reagents or testing materials, or if you just have questions about how the validation should be set up. So we'll obviously start the validation by determining and confirming the limit of detection. The FDA suggests using a clinical matrix. They prefer that to be VTM from lesion swabs. This may not always be possible or available, but efforts should be made to produce a similar matrix before spiking in the control material to determine the LOD. In the clinical evaluation, FDA is currently accepting contrived accuracy and precision samples. As testing ramps up and additional samples become available, this is likely one of the first requirements to change. If submitting an EUA, true patient clinical samples are required, or at the very least, deactivated cultured virus in contrived samples. So our validations all have utilized true patient samples to establish performance, but because those samples are very hard to come by, there's currently a little bit of wiggle room where truly synthetic contrived samples can be utilized. As the recommendations change, as more samples become available, it's currently a little unclear how the FDA is going to handle previous validations that have used fully contrived validations. But that's something that we'll have to figure out as the FDA moves on. So for inclusivity, currently in silico is accepted until more commercially available sequences are offered. So when making your primers, the FDA encourages both a monkeypox specific region. It's fairly popular right now to use a section of TNF. It has a lot of SNPs, and it can be, it's very helpful in specifically identifying the West African clade, as well as a highly conserved monkeypox virus target or non-variable orthopox target. So something that is less likely to mutate. So this is where you use something structural, like an envelope protein. That way you have both the monkeypox specific, but you can also detect, in the case of mutation, that the correct variant or the correct target is there. Now, regarding exclusivity and cross-reactivity, this is where you do wet testing against commercially available pathogens from common skin lesions. So you're going to want to see, cowpox or herpes 1, 2, measles, rubella, just to make sure that other things that may potentially mimic the presentation of monkeypox don't give you false positives. For interference, we'll look at both endogenous, whole blood. There's usually going to be a little bit of blood contamination in all of these samples, as well as exogenous wound material, saline wash, antiseptic, iodine, hydrogen peroxide, anything that would be present in the typical wound swab. And then finally, carryover and stability. So when it comes to actually running monkeypox in your lab, the CDC last week released updated guidance for how to prepare your lab to run the assay. And I know this is a fairly dense slide, but I thought it'd be a good reference. So feel free to take a screenshot or print this off. The full CDC recommendations can be found at the link at the bottom, or you can use a QR code to bring up their webpage. So to protect your staff and prepare your facility, this is the recommended workflow. For labs doing monkeypox testing, non-lesion specimens, so urine, blood, just standard chemistries, typically have a low viral potential in these types of samples. So standard precautions are appropriate, as well as BSL-2 confinement. So specifically for lesion specimens, this is where you expect a high viral load. So when possible, vaccinated individuals should be used to perform this kind of testing. So that's a smallpox vaccine within the last 3 years. When only non-immunized individuals are available, use additional PPE or BSL-3 practices within BSL-2 containment. So what does that mean? Using BSL-3 in a BSL-2 environment means everything possible is done within a biosafety cabinet. So this is everything from accessioning through extraction. So once you have an extracted sample, then you can go back to a standard BSL-2, but everything possible up until that point should be done in containment away from your staff. solid front gowns, double gloves, use of N95s, limiting personnel during specimen manipulation, and potentially directional airflow within the lab. Now, culture of lesion specimens is not likely something that this audience will lean toward, but for patients suspected of having monkeypox that need to be cultured for diagnostic purposes for something other than monkeypox, it's recommended that it can occur in BSL-2 facilities with smallpox vaccinated staff using BSL-3 practices. If the staff aren't vaccinated, it's recommended to use BSL-3 containment. So again, once the viral DNA is extracted using a validated extraction protocol, the viral DNA is no longer infectious and can be manipulated using standard BSL-2 containment facilities and procedures. They do specify validated extraction protocols. There are a lot of different methods out there for extracting genetic material. And if you're using something that may not completely lyse the virus, may not completely deactivate the virus, or wash the virus, then you want to continue being careful from there on out. But if you're using a validated extraction protocol that you know you have good lysis, you know that you're deactivating all the virus, then from the extraction on out, you can use BSL-2 operations. Now, vaccination of lab staff. There are two smallpox vaccines that are both approved for monkeypox. Past data suggests that they're about 85% effective in preventing monkeypox, even if given up to 4 days after exposure. Now, again, this is based on prior outbreaks. This is not a current outbreak, but the data seems to be holding. If given 4 to 14 days after exposure, vaccination can reduce symptoms, but it may not prevent the disease. So if an exposure does occur in your lab, if you have reason to be concerned, contact your local health department and they can help you facilitate vaccination. So if there are any questions, we can revisit them at the end of the talk. But for now, I'm going to pass the microphone back to John for a discussion of reimbursement and billing topics for monkeypox testing.
Thank you, Dr. Corti. Appreciate it. Really helpful. So as we look at sort of the business case, and is this something that makes sense for your lab? I thought in the polls, it looked like about 88% of you guys are set up to do PCR testing, and a little over half, 54% of you have been asked for monkeypox testing. So, you know, there's at least a reason to look at this, I feel. Also, U.S. leading in case count, right? So I feel like it's something that we need to pay a little bit of attention to. We've dug into the data a little in terms of reimbursement. CP2 code 87593 is the code that you would use if you're using probe or PCR technique to test for monkeypox, and the national rate for that is 5131. If you are in a MolDX MAC, which is Noridian, WPS, Palmetto, and CGS, which I believe is 28 of the states, you're going to reimburse at a lower rate, unfortunately, at 3509. Not much margin, if any, there to run it at that reimbursement. ACLA is advocating for a higher reimbursement of $77, so we'll see if they make any headway with that. And then, of course, commercial payers, it's going to be up to them, and there's too many of them to really know. But I haven't seen any real high rates coming from any of those commercial payers, but I'm not sure. There might be some opportunity there if you had a payer with a really good rate. We'll take some questions, and Deepak asked about reimbursement. Hopefully I'll be able to answer your question there. How can you get true patient samples to do a clinical correlation? We do have some access that you can buy them. Like I said, they might be expensive. We've seen them go all the way up to $5,000 per sample. I think you'll probably pay a little bit less than that, but that is something that is available with a little bit of searching, and we'd be happy to talk to you about that for our customers we're doing validations for. We get active in procuring those samples. Will it cover 100% to all Medicare and commercial? I think you're asking if there's 100% reimbursement or adjudication. I would say probably not. I think that Medicare will, commercial will be a different one, but I would expect it to be very high. I don't think that this is a high denial test. Where can we get inactivated viral stock? Eric, I don't know if you have any thoughts on that in terms of procuring inactivated viral stock for validation.
As far as the inactivated viral stock, and especially the true patient samples, finding a either a manufacturer or a testing facility that has them available is kind of a challenge at the moment. That's why the FDA is giving us a little bit of wiggle room on that. They've actually been fairly specific about their requirements for testing archived samples. So if there's a facility that has samples that they tested a month ago, they're actually not recommending that you use those for validation studies because the original labs will usually have to go back and retest those, and they're worried about testing capacity. So at least for the time being, for several reasons, they're letting you kind of get away from those aspects. As far as inactivated viral stocks for validation, I don't know any places offhand that have that. Someone did ask a little lower about the Zeptometrix recombinant stock, and if it's an appropriate reagent for validation that the lab director signs off on it, then absolutely that will be very appropriate for using in the validation study. FDA is giving us a lot of freedom in how we manage the validation within our own laboratories, which is nice. It allows additional testing capacity to wrap up quickly. They've learned a lot from COVID.
Great, very helpful. Jenny Harold is asking about RUOs, right? And so I think that you're going to see pretty much everything's going to be an RUO and would need to be used as an LDT. Yes, you can report out patient samples for an LDT as long as it's appropriately validated in your lab and signed off by your medical lab director. So that we can help with if you're looking to do a clinical validation on RUO to get it up to standards to be able to be used for clinical use, let us know. We're happy to help with that. So one of the other questions in New Jersey is, after we go through the validation, do we need to report to the State Department of Health to get their approval before testing or is reporting only to FDA sufficient? My understanding, Erica, and let me know if I'm incorrect, was that you needed to notify, but there was not an approval part. The approval of your medical laboratory director is going to be required to do the testing, but just a notification to the FDA. Is that accurate?
Yeah, that's right, John. So you have to notify the FDA within 5 days of initiating testing. But as far as the State Department of Health, your results have to go to the State Department of Health. And some states will want, I mean, at the current time, you're going to want to send all positive and negative results to the state. But those will follow standard reporting procedures for all your other reportables, just like they did with COVID and HIV, hepatitis, and those kind of conditions. So talk to your individual State Department of Health specifically with the reporting requirements. But as far as the ability to offer testing, the FDA just needs to be notified. As long as you do a full laboratory developed test validation and you're an approved laboratory for it, then you can go ahead and initiate testing.
Perfect. All right. And then just a quick plug for our next webinar, which is going to be on the topic of culture versus PCR. This is a hot topic right now. Everybody's got questions. If you live or work in a region that's governed by MolDX, then I'm sure you're familiar with the challenges of trying to make the case for PCR testing over culture, doing those technical assessments. And so we're going to bring in a few guest speakers and experts to talk about, just the clinical utility of that. Should we be doing culture or PCR? Which one makes more sense? How does the science play out? What does the reimbursement look like? What does the future look like as the market evolves? So I would encourage you to go ahead and sign up for that one. That's going to be on the 24th of October at noon Eastern time, and that'll be a free webinar as always. So I want to thank everybody for attending. I want to thank Dr. Corti for giving us his knowledge and researching the topic so that he could share with all of us what he had found. And please reach out if we can be of assistance here at Lighthouse. Thank you all.